Good morning, everyone, and thank you for being here today. I'm Dr. S.K. Singh, and I'm pleased to welcome you to this presentation. Today, we will be discussing the stability requirements for new drug substances and new drug products. Over the course of this session, we'll explore the key standards and considerations necessary to ensure drug stability. We'll cover regulatory expectations, testing methods, and best practices. I look forward to sharing valuable insights with you all. Let's get started. Let's take a closer look at the stability requirements. The primary objective here is to define the stability data package for a new drug substance or drug product. This data needs to be comprehensive enough to support registration applications across different climatic zones, specifically zones I, II, and IV. In essence, we're establishing what information is necessary to demonstrate that the drug remains stable and effective throughout its shelf life, regardless of the environmental conditions it may encounter in these zones. Moving on to the scope, this involves the specific information that must be submitted when applying for registration of new molecular entities and their associated drug products. This means that the stability data isn't just a formality. It's a critical component of the registration process that ensures the product's quality, safety, and efficacy are maintained throughout its life cycle. Understanding these requirements helps us create a robust data package that facilitates smooth registration and ultimately supports the successful launch of new medicines in various markets. The purpose of stability testing is to gather evidence on how the quality of a drug substance or drug product changes over time. This involves examining the effects of various environmental factors such as temperature, humidity, and light. By understanding these influences, we can determine an appropriate retest period for the drug substance or establish a shelf life for the drug product. Additionally, stability testing helps us recommend suitable storage conditions to maintain the product's quality throughout its life cycle. These principles are fundamental to ensuring that medicines remain safe and effective until their expiration date. The climate varies significantly across different countries around the world. Because of these differences, it's essential that stability studies for drug substances and products are conducted in accordance with the specific climatic conditions of each country. According to the ICH guidelines, the global climate is categorized into 5 distinct zones to help guide these stability studies. The first zone is the temperate zone, which generally includes regions with moderate temperatures and humidity levels. Then we have the Mediterranean or subtropical zone, characterized by warm, dry summers and mild winters. The hot, dry zone is next, which experiences high temperatures with low humidity. Moving on, we have the hot, humid or tropical zone, where high temperatures are coupled with high humidity levels. Finally, the hot, higher humidity zone is similar, but typically involves even greater humidity, often found in certain tropical or subtropical regions. Understanding these zones is crucial because it helps us tailor stability testing conditions. This way, we ensure that our drug products remain effective, safe, and of high quality throughout their shelf life, regardless of where they are stored or used. Recognizing the climatic zone of a specific country allows us to set appropriate testing parameters, ultimately supporting global health and regulatory compliance. Let's take a look at storage conditions, as they vary according to different climatic zones. Understanding these conditions is crucial because the environment directly impacts the quality and longevity of stored products. In different climatic zones, whether it's tropical, temperate, or arid, the storage requirements differ significantly. For example, in tropical zones, high humidity and temperature levels demand more robust moisture control and ventilation systems to prevent spoilage. Conversely, in temperate regions, storage conditions might focus more on temperature stability to avoid freezing or overheating, which can damage sensitive items. In arid zones, controlling dust and maintaining appropriate humidity levels are key, as dry environments can lead to product desiccation or cracking. The main takeaway here is that storage conditions must be tailored to the specific climatic zone to ensure optimal preservation. This concept ties back to our overall theme of adapting storage practices to environmental factors, emphasizing that a one-size-fits-all approach isn't effective. By aligning storage conditions with the local climate, we optimize product quality and reduce losses, which is essential for efficient supply chain management. Let's take a closer look at storage conditions as per different climatic zones. This is a crucial aspect because the environment in which we store goods can significantly impact their quality and longevity. Different climatic zones, such as tropical, temperate, or arid, each have unique temperature, humidity, and sunlight levels. So, understanding these differences helps us determine the best storage conditions. For example, in tropical zones with high humidity, we need to focus on controlling moisture to prevent spoilage or mold growth. Conversely, in arid zones, maintaining proper humidity levels is essential to avoid drying out products. Temperature control is equally important across all zones. Many items require stable temperatures, and fluctuations can lead to deterioration or loss of quality. By tailoring storage conditions to each climatic zone, we not only preserve the integrity of the products, but also optimize shelf life and reduce waste. So, recognizing the specific needs of each zone allows us to implement appropriate storage strategies, which ultimately supports the overall goal of maintaining product quality throughout the supply chain. Now, when we talk about the batches for the drug substance and drug product, it's important to understand that we typically work with at least 3 primary batches. These batches are produced using the same drug substance, formulation, and container closure system that we plan to use for the actual production and marketing. This consistency helps ensure that what we produce in these batches accurately reflects what will be on the market. For drug products specifically, we generally prepare 2 pilot scale batches. These are usually about 1/10 of the full production scale, or around 100,000 tablets or capsules. The goal here is to simulate the manufacturing process on a smaller scale, so we can evaluate and troubleshoot, making sure everything runs smoothly before full-scale production. In addition, we might produce one smaller batch, perhaps around 10,000 tablets or capsules. This smaller batch serves as an additional check, providing more data on consistency and process robustness. When selecting these batches, the key is to cover the typical manufacturing conditions and ensure they are representative of the final product. This approach helps us confirm that the process is reliable and that the product quality remains consistent across different batches. By carefully choosing and evaluating these primary batches, we set the foundation for a successful transition from development to commercial production, ultimately supporting our overall quality and regulatory strategies. Now, let's talk about stability testing. It's essential that we conduct this testing on the actual drug substance or product that will be packaged in the container closure system we plan to use for storage, distribution, or marketing. This includes, of course, the primary packaging, but also any secondary packaging and labels that are part of the final product. The idea here is to ensure that the stability data truly reflect the conditions the product will face in real-world scenarios. Additionally, it's worth noting that any studies we've already done on the drug product outside of its immediate container, say, in other packaging materials or different conditions, can be quite helpful. These studies can support the stress testing process or serve as additional background information. They help us build a comprehensive understanding of how the product behaves under various stresses and conditions. When we talk about the container closure system specifically, we want to make sure that the entire system is compatible with the drug product and maintains its stability throughout its shelf life. This means thoroughly evaluating the packaging materials and their interactions with the drug. In essence, our goal with stability testing is to confirm that the product remains safe, effective, and of high quality until the end of its shelf life, and using the right container closure system is a critical part of that process. When we talk about stability studies, it's essential to focus on testing those attributes of the drug product that can change during storage and could impact its quality, safety, and efficacy. These are the key factors we need to monitor to ensure the product remains effective throughout its shelf life. The testing should be comprehensive, covering physical, chemical, biological, and microbiological attributes as appropriate. This means we look at things like appearance, potency, purity, and microbial contamination, depending on the nature of the drug. Now, when it comes to defining the shelf life acceptance criteria, these should be based on all the available stability data we have. This often involves extrapolating data to predict how the product will behave over time, especially when real-time data might be limited. Ultimately, the goal is to establish clear, scientifically justified specifications that ensure the drug remains safe and effective for the intended duration. This approach helps us support the overall quality assurance of the product throughout its life cycle. Let's now talk about testing frequency and storage conditions. These are critical factors in ensuring the reliability and integrity of our samples or products. First, testing frequency refers to how often we need to perform quality checks or assessments. It's important to strike a balance. Testing too often can be resource-intensive, but too infrequent testing might let issues go unnoticed. We need to set a schedule based on the stability of the product and the risks involved. Next, storage conditions are equally vital. Proper storage ensures that samples or products don't degrade over time, which could compromise their quality or safety. Factors like temperature, humidity, and light exposure all play a role here. Maintaining consistent storage conditions helps us preserve the integrity of what we're working with. Connecting these points to the overall theme, consistent testing and appropriate storage are essential parts of quality control. They help us catch potential problems early and maintain the standards we aim for. By paying close attention to these details, we ensure that our processes are reliable and our results are trustworthy. When it comes to requirements for submission, the key thing to remember is that you need to have at least 12 months of long-term testing data and 6 months of accelerated testing data available when you're ready to submit. This means you should have a solid timeline of data showing how your product holds up over time under normal conditions, as well as under more stressful, accelerated conditions to predict long-term stability. Now, if any significant change happens during the accelerated storage condition, say before the 6 months are up, you'll need to include intermediate testing data, at least 6 months' worth. This helps demonstrate how the product is evolving during storage and ensures safety and efficacy are maintained. Speaking of significant change, let's clarify what that means. Generally, a significant change is considered a 5% change in the assay from the initial value. So, if your active ingredient concentration drops or increases by more than 5%, that's a red flag. It also includes any degradation product exceeding its acceptance criteria. Basically, if harmful or unacceptable degradation products form beyond the set limits, that's significant. Additionally, failure to meet acceptable criteria for appearance, physical attributes like hardness, or functionality tests also counts. For example, if a tablet becomes too soft or hard, or if it no longer disintegrates properly, these are critical issues. And finally, if the product fails to meet the acceptance criteria for pH or dissolution in just 12 dosage units, meaning even a small batch shows problems, that's also considered significant. All these points are crucial because they ensure that the product remains safe, effective, and of high quality throughout its shelf life. So, thorough testing and careful monitoring of these parameters are essential parts of our submission process. Let's talk about storage conditions. This is a crucial aspect because how we store our products can significantly impact their safety and efficacy. First, proper storage conditions help maintain the stability of the product over time. This means keeping the right temperature, humidity, and light exposure is key. For example, some medicines need to be stored in a cool, dry place, while others might require refrigeration. It's also important to follow the manufacturer's instructions carefully. Deviating from recommended storage conditions can lead to degradation or loss of potency. This is especially relevant in the context of overall pharmaceutical quality and ensuring patient safety. Additionally, consistent storage conditions help prevent contamination and spoilage. Proper labeling and organized storage areas make it easier to monitor conditions and avoid mix-ups. In the bigger picture, maintaining optimal storage conditions is a vital part of the entire supply chain. It ensures that the products delivered to patients are safe, effective, and of high quality. So always pay attention to these details. They truly make a difference. When we have long-term stability data on primary batches, but that data doesn't fully cover the proposed retest period or shelf life granted at approval, we need to make a clear commitment to continue the stability studies after approval. This way, we can firmly establish the appropriate retest or shelf life period. Now, if the submission includes data from production batches, the requirements depend on how many batches are provided. If there are at least 3 production batches, then we commit to continuing the long-term stability studies until the proposed retest period or shelf life, and we also need to perform accelerated studies for 6 months. If fewer than 3 batches are submitted initially, then we commit to studying the remaining batches until we reach a total of 3, again covering the long-term period and including accelerated studies for 6 months. And if no production batches are submitted at all, then our commitment is to place the first 3 production batches on long-term stability testing through the proposed retest period or shelf life, with additional accelerated studies for 6 months. This approach ensures that we are thoroughly supporting the stability profile of the product, which is a key part of the overall quality assurance process. If you need any further support, please don't hesitate to reach out to us. We're here to help you succeed and answer any questions you might have. Thank you so much for your time and attention today. We really appreciate your interest and engagement. Remember, support is always available, so feel free to contact us whenever you need assistance. Thanks again, and have a great day.